Assalamualaikum and hello everyone. We are from Group 1 from the SBB 3033 Principal in Microbiology course. First of all, let me introduce all of the group members, which is Muhammad Irfan Irshaduddin bin Muhammad Zainuddin, Nur Syafiqah Aisyah binti Muhammad Kasri, Muhammad Syakir bin Muhammad Kazdi, and last but not least, Muhammad Syarizan Aizat bin Syamsul Puan. Welcome to our SULAM Photovoice project titled Invisible Life, Microbial Wonders in Group C. Before we dive into this project, we need to first know what is microbes are. Microbes are everywhere, in the water we touch, the leaf we pass, and even the air we breathe. Yet, most go unseen. This project explores the hidden microbial life on our campus using real microbiological technique by collecting environmental samples and applying what we learn in the lab. We'll uncover a world full of living organisms, tiny but powerful. This journey not only reveals their presence but also their importance to health, hygiene, and the environment we share. Before our project began, we first attend an online briefing via Google Meet where we receive guidance on how to connect our lab work with real-world applications and how to complete this Photovoice project. We began our project by selecting Campus Sultan Abdul Jalil Shah, UPSI, as the venue. Our project location located around Institusi Peradaban Melayu UPSI was chosen because it is a shared public environment where microbiome presence can affect the health and hygiene of the community. There are three samples that we all agree and chosen from around this location, namely leaf A, leaf B, and water. Throughout this project, there are several objectives that we need to achieve before the end of this project, which is to apply technique in microbial cultivation, to transfer single colony bacteria, to perform technique of smear preparation, to observe potential microbial chemotaxis in response to chemical agent, to isolate pure colony by strip plate method, to understand the chemical and theoretical basis of biological staining, and lastly, to measure microbial growth using enumeration method.
Introduction to microbiological techniques. Before we investigate microbe in wild, we begin in the lab, which is practicing aseptic techniques that are essential to safety and accurately handle microorganisms. This includes tracking plate to separate colony, transferring microbe to broth to promote growth, and inoculating step agar to study bacterial movement and oxygen need. This foundational skill equipped us for field sampling and taught us how to avoid contamination. This early hand-on experiment helped us feel confident using microbial tools and build our understanding of how to work with living organisms that cannot be seen with naked eye. Disinfectant testing. One of the most fascinating things we observed was chemotaxis, which is the ability of microbes to sense chemical and move toward or away from them. By placing substance on agar plate, we saw directional microbial growth, showing that bacteria can respond to their environment. This moment challenged our idea that microbes are passive. In reality, they behave with purpose, survival instinct, and even preference. This behavioral trait deepened our understanding on how infections spread and how microbes find nutrients in their environment. Microbial development. Once we plate our sample onto nutrient agar, the invisible begin to appear. Over the next few days, bacteria form visible colony, each one representing a single organism that had multiple many times. Using colony forming unit (CFU) techniques, we estimate the number of microbes present and studied how they differ in shape. This physical variation reflects the diversity and complexity of microbial life. Even within a single sample, every plate tells a different story.
And now we will proceed to the specimen staining. By using differential techniques such as simple staining and gram staining, we were able to visualize bacteria. at the microscopic level in our eyes alone could never achieve. We identified various bacteria shapes, including cocci, bacilli, and vibrio, observing not only their form, but their arrangement. The staining process also illuminated the cell wall properties of bacteria. Gram-positive organisms retain the crystal violet stain and appeared purple under the microscopy, while gram-negative bacteria appeared pink color. Next, we will get to the community connection. Microbes are everywhere, and most are harmless or even beneficial, but some can pose risks if not understood or managed. By studying environmental microbes on our campus, we learn how important it is to clean shared spaces, handle food safely, and use the right disinfectant. These experiments made microbiology feel deeply connected to our daily lives and our community. Whether in cafe, classroom, or garden, microbial awareness helps us protect each other and make informed decisions about hygiene and our health. And lastly, we will address about the reflection throughout the SULAM project. From natural samples to microscopic worlds, this journey showed us that microbiology is everywhere. It's unseen, but it's so really important. That's all from us. Thank you, and thanks for watching.